Welcome to Update from ASCO. We're very excited to have this webinar today. During this seminar, all participants are going to be in a listen-only mode, and there will be a question and answer session at the end of the presentation using questions submitted through the Q&A feature. Please remember the information provided in this web seminar is not a substitution for your physician's guidance and care. My name is Sarah Rothschild. I'm the Executive Director of the Life Raft Group, and I'd like to introduce our presenter today. Dr. Herbert Loong holds conjoint appointments of Clinical Associate Professor in the Department of Clinical Oncology and Deputy Medical Director of the Phase 1 Clinical Trial Center of the Chinese University of Hong Kong. He is also the current and founding convener of the Prince of Wales Hospital Adult Sarcoma Multidisciplinary Team, and he obtained his medical degree with a distinction in surgery from the University of Hong Kong in 2003. He's the recipient of the European Cancer Congress Fellowship Grant in 2013, ASCO's Annual Meeting Merit Award in 2014, and the Hong Kong College of Physicians Young Investigators Award in 2014. And we also know that he does so many other things beyond that. So we're very excited to have him here today to present updates from ASCO. Thank you for joining, and I'm going to hand it over to him.
Thank you very much, Sarah, and it's a pleasure to be here again, and certainly good evening, good morning, wherever you are. I'm going to try to share my screen first. Let's try getting that done. Hold on. Okay, let me figure this one out. Share screen. Just bear with me for a little bit. Oh.
No problem.
Okay.
In the meantime, if anybody wants to start putting questions into the Q&A feature, feel free to do so.
Okay, are we able to see the screen?
Yes.
Yes, and that's the full screen, right?
Correct.
Okay, good. Okay. Thanks, Sarah. And again, it's a real pleasure to be here. And it's also a real pleasure to be able to be given the opportunity to highlight some of the abstracts that were presented at ASCO just earlier this month. So my name is Dr. Herbert Loong. I'm a clinical associate professor in the Department of Clinical Oncology at the Chinese University of Hong Kong. And in the next 30 minutes or so, I'll present to you some of the key abstracts on GISTs that was also presented at ASCO earlier this month. So these are my disclosures. And also a disclaimer, the copyright of the slides being used today are actually belong to the original authors and or ASCO and are being used by courtesy for this academic as well as patients educational event. The choice to discuss which abstracts was made on my own assessment of their academic merit and the impact to the GIST community, and by no means necessarily reflect the views of ASCO, of which I'm currently serving as a scientific committee member of. So I've selected three main abstracts that were highly rated at ASCO, which were also given oral podium positions to be presented and to share with the community here today. This includes the STRATA-GIST-1 trial, which is the first in-human phase 1 study of the compound IDRX42 in patients with metastatic GIST tumors resistant to prior treatment with tyrosine kinase inhibitors. And this was presented by Professor Shofsky. The second abstract I'll discuss is the phase 1 study of NB003, which is a broad-spectrum KIT PDGFR alpha inhibitor in patients with advanced GISTs, presented by Dr. Lee on behalf of his co-investigators, as well as the third abstract titled Updated Efficacy Results of Overembatinib, or HQP1351, in patients with TKI-resistant sdh-deficient GISTs and paraganglioma, and this was presented by Dr. Chiu on behalf of his colleagues in the oral abstract session as well. So going back to with a little background, and certainly our community here is well aware of the fact that when we talk about GISTs, we're not really talking about just one singular disease, but we also know that throughout the treatment journey, especially in the situations where it's advanced metastatic setting, where we're giving the different targeted therapies, the GIST tumors tend to then potentially behave differently. And this is due to some of the underlying changes in terms of development of mutations in the tumors after initial exposure to different tyrosine kinase inhibitors. So I borrowed the slide from my good friend and colleague, Dr. Jason Siklik, on the slide on the left to really illustrate to the community that in terms of our treatment approach to GISTs, I think even as of today, with the increasing number of drugs available, we still take a relatively pragmatic approach, where in the initial onset, in situations where there's advanced or metastatic disease, we'll start off with imatinib or Gleevec, where it actually gives, for most patients, a fairly good longer term of disease control. But once there is evidence of disease resistance, we may move on to second line, as well as third line, as well as fourth line. And as you can see, as we go progressively down the treatment pathway and down the different lines, the amount of time of control is actually relatively less. And this, again, has to do with the fact that we know that GIST is not one singular disease, and certainly there are different molecular alterations in the GIST tumors, and this can also be induced by the use of various treatments. And as you can see here in this preclinical experiment diagram, essentially it just shows the different type of the tyrosine kinase inhibitors, and you can see that there are some situations where, say, imatinib or sunitinib is very useful, but actually is proven to be resistant in these secondary mutations. But the characteristics of all these TKIs are actually quite different. We also know that with increasing evidence of molecular profiling, we also know that in actual fact, the different drugs may have different activities in different situations. For example, in this particular study that was presented as an ASCO virtual plenary, we know that, for example, in patients who have tumors with exon 11 mutations, together with the resistant mutations of, say, exon 17, 18, in actual fact, they respond very well to ripretinib, but not good at all to sunitinib. So we're starting to have an increasing variety of drugs made available, and potentially it also means that we may personalize the treatment approaches based on the molecular profile of each of these patients' GIST tumors. Now, talking about the first abstract, and this is presented by Dr. Shofsky in the oral session, which is a compound known as IDRX42. And this is a potent, highly selective pan-mutant KIT tyrosine kinase inhibitor, and it has a broad coverage of disease-relevant KIT mutations, and with excellent enzyme and cell-based kinase selectivity. In actual fact, I think I presented to the GIST community some years ago, maybe 2 years ago, when this abstract was first presented as a trial in progress poster at ASCO 2022 by Dr. Suzanne George. In actual fact, this abstract, or this particular compound, was also highlighted in the recent CTOS meeting by Dr. Shofsky, where in November earlier last year, he presented the data in Dublin, where in actual fact, we were able to see the fact that there is very good response in a large variety of patients, with a clinical benefit rate of 68%, meaning that 68% of the patients who were in the clinical trial on the drug had at least no further growth of the tumor and possibly shrinkage of the tumors whilst on this particular compound. And essentially, it also shows the fact that this particular compound is active in a preclinical setting, at least in the laboratory, against a variety of commonly found GIST-associated mutations, both in the primary mutations, as well as some of the resistant mutations that we see as well. Now, going into a little more detail about this particular study, this study recruited patients with metastatic and or unresected GIST, with known KIT mutations who had prior use of imatinib and good performance status. And essentially, this is a phase 1 study where the main objective is to look at the safety and the toxicity of the particular drugs. And as you can see here, it's gone through a dose escalation process, where in actual fact, a small number of patients, n equals to 3, is actually initially treated at a lower dose. And when it's deemed to be safe, in actual fact, the trial then goes up to a higher dose, and the dose is escalated until there potentially is a situation of toxicity, or until the fact that they feel that it is actually efficacious. Now, in actual fact, they've gone through the process of escalating all the way up to 1,200 milligrams per day. But they have actually decided to go on to expansion at a slightly lower dose. And the presentation that Dr. Shofsky made at ASCO is mainly about this phase 1 dose escalation portion. And there's a plan to then subsequently expand the study into different patient groups as well. Now, about this phase 1 dose escalation portion, in total, there were 73 patients that were recruited into the trial. As you can see here, it includes patients who are typically the patients that we would see in clinic in terms of characteristics. Do note the fact that most of these patients, so 70% of these patients, have had at least 3 or more lines of prior treatments. So typically they would have gone through imatinib, sunitinib, as well as regorafenib. And in actual fact, most of these patients had exon 11 mutations, as we would expect. Also, in actual fact, 29% of these patients had exon 9 mutations. Now, this was the data that was presented by Dr. Shofsky. And as you can see here, this is what we call a waterfall plot, where we plot out the changes in the diameters of the tumors that we see on the CT scan. Anything that points downwards indicates the fact that the tumors are shrinking in size because it's the sum of the diameters. And anything that's going upwards means that it's actually increasing in size, so resistance to treatment, and means that the drug is not working. And as you can see here, I mean, just as an overall impression, most of the bars are pointing downwards, indicating the fact that most of the patients are actually responding to treatment whilst they're on treatment. The different colors represent the different doses that were used in the dose escalation. And given the fact that this is relatively rainbow-like with many colors intercalated with each other, what you can see is the fact that most of the patients actually benefited from treatment regardless of whether or not they got a very high dose or a low dose. And the plan actually is to expand, I think, at the 600 milligram dose level, which is the purple bars. This is actually the more important aspect here, the box that's been highlighted in blue, which indicates the different types of KIT mutations that each of these patients had in their tumors. For example, in this one, which is most furthest on the right, which had the best response, this patient actually had tumors with known KIT exon 11 mutation, as well as exon 17 resistant mutations. And having these two mutations, together with 5 prior lines of treatment in terms of different tyrosine kinase inhibitors, potentially entering into prior clinical trials as well, this patient also generated a very good response with about 65% in shrinkage of the sum of the longest diameters of their tumors whilst on the study drug. Now, this on the right is what we call a swimmer's plot, and each of these bars represent an individual patient. It's called a swimmer's plot because it's like looking from above, looking at a swimming pool from above. Each of these people are swimming, and each of these are lanes. And you can see that any of these with arrows means that at the time of data analysis, the patient was still on treatment, and the length of the line represents the amount of time that they're actually on treatment. And you can see that most of these patients have been on treatment for a long period of time or continues to be on treatment. And this is also very encouraging because it means that in terms of the side effect profile as well as the efficacy, there is a very good promise with this particular drug. So the key take-home message is the fact that there is preliminary efficacy, and I would say it's preliminary because it's a small number of patients, but in a molecularly diverse as well as a heavily pretreated group of patients with very encouraging duration of responses. And this is actually, again, a very promising drug, which we will continue to receive information at upcoming meetings as well. Now, there are just 2 quick case vignettes that Dr. Shofsky shared at his presentation, which includes this 60-year-old man who was treated at a slightly lower dose of the drug, where he has had prior multiple lines of treatment. And in actual fact, after treatment, 4 weeks, in actual fact, the tumor has shrunken quite significantly in size, as you can see here. And this is also a plot of the liquid biopsy, which looks at the circulating tumor DNA within the body. And in actual fact, they were able to document the fact that the circulating tumor DNA went down quite quickly after treatment and continued to be low throughout the treatment journey. On the right is actually another patient who has a tumor right here with one prior line of treatment. And after treatment with IDRX42, there is a metabolic response in terms of the tumor as well. Now, whenever we talk about drugs, we always have to talk about the safety profile and the potential side effects as well. And it was commented the fact that it had a relatively favorable safety profile. And most of the patients who had side effects or toxicities mainly had what we call GI toxicities, which really has to do with, say, diarrhea or nausea or vomiting. In actual fact, 68% of the patients had some diarrhea, with 51% of them having grade 1, but there were 4% which actually had severe diarrhea, as well as half of the patients having nausea and about a third of patients having some vomiting. These are important aspects, which means that we do need to make sure our patients who are on these drugs have these side effects well taken care of. But it's also reassuring to note the fact that there is a very low rate of dose modifications. So many of these patients who actually had these side effects did not necessarily need a change of the dose. Only 6% of the patients had reductions, and 9% of the patients had to have the treatment interrupted because of any of these side effects. So aside from that particular abstract, another interesting abstract that's also in the GIST space was this that was presented by Dr. Lee and colleagues of another compound known as NB003. This is another broad spectrum KIT or PDGFR alpha inhibitor in patients with advanced GISTs. And again, by way of background, you've seen this diagram already, but this is a slide that was used by Dr. Lee indicating the fact that NB003 is a compound which actually has at least preclinical activity against a large variety of mutations in GISTs found either as primary mutations or secondary mutations. It also seems to have good effect on PDGFR alpha, and of course in the PDGFR alpha D842V specific mutations as well. Now, again, this is a phase 1 study, so they are looking at the early safety signals of this particular compound, where again patients were recruited and then the dose of the drugs were escalated, and then subsequently upon finding the actual dose to go for expansion, they would then expand into the different cohorts and groups. Now, again, they were presenting the dose escalation data, so there are a smaller number of patients, and a lot of these patients are treated, as you can see, at different doses. This is just showing the demographics, which indicate the fact that, again, most of these patients have had a large or high number of prior lines of treatment. And in terms of side effects, and this was also presented at the meeting, it seems as though the side effects are somewhat different from IDRX42. The more common side effects have to do with some of the laboratory investigations, including what we call blood creatine phosphokinase, or CPK, which is actually an enzyme that's typically found in muscles. And some of these actually had elevation in the patients. And then there were also issues with blood counts, including neutropenia, as well as anemia. And these are common side effects that we see in some of the chemotherapy drugs. Face edema or facial swelling is something that's also seen. This is probably what we call a class effect. Most of the drugs that affect KIT have this similar side effect profile. We see this in imatinib. We also see this in ripretinib as well. Now, I think in terms of response, again, these is very early days. These are a very small number of patients, but you can see that in all the patients that's been recruited, the response rate was documented at 26.2%. And specifically for patients who have had less prior treatment, more of these patients actually responded. So 100% of the patients who had 2 prior lines of treatment responded. But note to the fact that there were only 2 patients in this group. But I think overall it means that there is a signal of activity of this drug in GIST patients. And this is a plot known as a spider's plot, where we actually look at each color is each line is each individual patient in the clinical trial, And again, this plots the diameter of the tumors whilst they're on treatment. And as, for example, here, as the patient starts the drug, the diameters of the tumors have shrunken in size, so much so that it is what we call a partial response, whereas there's a shrinkage of more than 30% of the tumor. And at 8 weeks, in actual fact, there's a shrinkage of the tumors to a total of about 75%. But by week 16, for this particular patient, for example, we're seeing growth of the tumors again. And essentially the length of these lines, which shows and the trajectory of these lines shows how effective the drugs are for each of the individual patients. On the right side also is the swimmer's plot for this particular compound, again indicating the fact that quite a fair number of patients were continuing on treatment at the time of data cutoff for presentation. Now, again, this is another plot showing the response rates of these particular patients on the drug. And you can see, again, at the various doses in different colors, you see patients who actually have responses. And most of these patients actually had some shrinkage of the tumor, with a small number of patients actually had more than 30% shrinkage. What's also interesting is, again, you also have patients who have a variety of primary as well as resistant KIT mutations and multiple lines of treatment as well. So again, this is a compound or a drug which is active in patients who have been pretreated with prior tyrosine kinase inhibitors. Now, overall, if you look at these two studies, which really is aimed to expand the amount of mutations that the drugs can be active against, the approach is actually quite similar to an active phase 3 clinical trial at the moment, known as the Cogent PEAK study. The PEAK study is a global randomized phase 3 study of a drug, a novel compound known as bezuclastinib, together with sunitinib, which is our standard second-line drug in patients with GIST. And bezuclastinib is a compound which actually has a specific activity on the typical GIST primary mutations, as well as exon 17 and 18, which sunitinib unfortunately doesn't have coverage of. And the whole premise of this particular clinical trial is really to combine the two agents, so therefore you get good coverage of both the primary mutations as well as all the well-known secondary mutations, and therefore, in actual fact, hopefully having a broad spectrum activity, and then also then reducing the risk of having disease resistance and hopefully having better disease control. Now, the trial is ongoing. It's a global trial that's actually recruiting at this very moment, and hopefully my understanding is recruitment is near completion. So hopefully we'll be able to see some of the data shortly in some upcoming conferences. Now, when we combine two drugs, the biggest worry, of course, is whether or not it increases side effects. Based on some of the earlier data of this combination that was presented by Dr. Somaya at CTOS earlier last year, again, it seems as though the gastrointestinal side effects seems to be most common, with diarrhea as well as some nausea. But I would say these are actually relatively low grade or small in number in terms of the side effects. So potentially this is another option which in terms of the drug development process is in a more mature state, because it's already on a phase 3 study. So we look forward to seeing some of these results. So the logical step forward, based on these abstracts, really is for any approach to trying to broadly cover both primary and secondary mutations, the paramount importance is really to look at the toxicity profile as well as the treatment tolerance for our patients. And now, if these are really effective treatments, is it possible to move this particular approach to an earlier line setting? So traditionally, we treat patients with imatinib. It has a very good response for the large majority of patients, but we know that it doesn't, for example, cover typically exon 17, exon 18, nor is it good for exon 9 mutated tumors. Now, if we can have a broad coverage up front, would that actually help reduce the risk of resistance and therefore hopefully increase the duration of response? In actual fact, this is an approach that we typically do now for lung cancer. You may have heard of a drug known as Tagrisso or osimertinib, and this was a newly novel drug developed to cover extreme resistant mutations, and we've now already moved that into the first line setting. So this is a potential approach that, as us in the GIST community, can consider doing as well. but there are potential drawbacks because it means that we may be diverting tumors which actually develop resistance because of various KIT mutations into other types of mutations of which we don't have drugs for. So I think we need to learn a bit more in this before we actually consider moving this into the first-line setting. But I think it's also important for us to note the fact that we now recognize that, again, GIST is not just one disease. There's a variety of GISTs based on the molecular profile. Typically, when we talk about GIST, we talk about KIT, we talk about PDGFR alpha, and the two combined already accounts for about 85 to 90% of all GIST in our community. Having said that, there are also the remaining 10%, which is associated with SDH deficiency as being one of the alterations, and classically was known as wild-type GIST. Now, we don't know that there are some rarer GIST which is not associated with KIT, but also associated with known alterations. As I mentioned previously, sdh deficiency is one of them, but there are also other mutations, including, say, NTRK fusions, BRAF mutations, and NF1, etc. So I think now we have more drugs available. It is important for us to delineate which mutations the patients have in their GIST tumors, and molecular profiling is, I think, essential for all newly diagnosed GIST patients. And the next abstract I'll present is about sdh-deficient GIST, which is a smaller group of GISTs. But just to really give a shout out to the Life Raft Group for putting together an abstract earlier at ESMO last year, really looking at the amount of molecular profiling that's been done throughout the years. And what they have noticed is actually between the years 2017 and 2022, because there's increasing use of molecular profiling, more SDH mutation GIST patients were identified. And I think this is actually quite important because we know that these tumors do behave differently. There are actually no specific drugs available which is effective in this group of patients yet, but we'll talk about a compound that shows very good promise. But I think it's important again to identify which patients would benefit from whichever treatment. Now, going into SDH-deficient GISTs, I would presume actually the community probably knows a bit better than me, but essentially we know that this is a subgroup of GIST, or you could actually even classify it as a completely different disease because of the fact that molecularly, or at least in terms of its genomics, it's actually quite different from our typical KIT mutated or GISTs. Now, SDH-deficient GISTs typically occur in the younger age group and more in females, typically in the stomach, and when they first present, they're typically multifocal as opposed to just having a singular tumor. It's associated with various inheritable syndromes, such as the Carnes-Straktakis syndrome, or what is also known as the Carnes triad, with also the development of pulmonary chordomas, as well as paragangliomas. And what's more interesting is the fact that the imatinib, which is a typical first-line drug for GIST, is not effective in this particular group of patients. And in some of the prior clinical trials with, say, sunitinib or regorafenib, there are some small amount of responses. So some patients have shrinkage of the tumors, but again, probably not because of the fact that these drugs act on KIT, but there are other off-target effects, including, say, VEGF, which may play a role in leading to the shrinkage of these tumors. But I think it's suffice to say that these response rates are relatively low, and there are no good treatments for SDH-deficient GIST in the advanced or metastatic setting. Now, Dr. Chiu and colleagues presented the data of a drug known as overambatinib. And overambatinib is a third-generation tyrosine kinase inhibitor, which is actually currently already approved in mainland China for the treatment of chronic myeloid leukemia in patients who actually had disease breakthrough or resistance to first-line treatment. The preliminary data in GIST was actually presented in 2022, also at ASCO, which showed the fact that the patients who are in this clinical trial, these patients here, again, in the swimmer's plot, in the light blue, are patients with SDH-deficient GISTs. And you can see that a lot of these patients had longer duration of responses. Whereas in actual fact, the patients with typical GIST-associated alterations, including KIT exon 11 and other KIT alterations, actually had shorter durations of treatment. Now, in terms of safety profile, it seems as though, again, this is a drug which is relatively safe. The most common side effect actually is pyrexia, which is fever, which means that by taking this drug, these patients actually develop fever, but many of them were relatively low grade. There were also some issues with liver function, but these were kept at what we call grade 1 and 2, and very small numbers had higher grades. So in general, this would be considered a relatively safe drug. The median treatment duration was about 15.6 months. And only 2.3% of the patients experienced what we call a serious adverse event, which typically means that it required hospitalization, but there were also no deaths reported in this particular trial. And this is the efficacy results, and this is actually quite impressive, noting the fact that these are patients with sdh-deficient GIST tumors, which traditionally there are no good treatments for. And you can see that the majority of the patients actually had at least stabilization or shrinkage of the tumors, with these patients here having more dramatic responses and documented as what we call a partial response. What is even more impressive is the swimmer's plot, where at this time of data cutoff, you can see that patients are continuing on treatment. These green arrow shows that they're continuing, and the longest duration of treatment right now is a patient who's been treated for 42 cycles, who has stopped, but there's this one patient who's treated for 40 cycles, and many of these continues to be ongoing. Now, I think based on this data, there is clear signal of activity in sdh-deficient GISTs, but it's also clearly ineffective in KIT-driven GISTs. In actual fact, Dr. Chiu previously shared some information noting the fact that if you have a GIST which did not have sdh deficiency, in actual fact, half of the patients actually progressed by the time of the first imaging scans. But for the patients who were benefiting, there's a long duration of ongoing treatment response. And in terms of the side effect profile, it's actually been described in this particular trial. Because the drug is already approved in chronic myeloid leukemia, we actually have a large number of patients who's been exposed to this drug. So the toxicity profile is actually very well described as well. There's a plan to bring this drug to a phase 3 clinical trial in China to seek approval. But I guess the question is, do we actually need further randomized study in this situation, given the fact that this is a rare disease group and we actually have the safety data already available? Maybe it's not necessary to do a large randomized trial, whereas in actual fact, by collecting all this data that we have, as well as collecting the data in terms of the side effect profile from the CML experience, we may already be able to build a large enough portfolio for regulatory approval for this particular drug. So this drug actually already has breakthrough therapy designation in China. My understanding is there's a plan to bring this to phase 3, but a single arm open label phase 3, and we could talk about what that really means later during the Q&A. But in any case, I think what we really need to do is make sure that we have the evidence available, and hopefully we'll be able to then have this drug approved and then bring this drug to patients, because there are otherwise no good treatment options for patients with sdh-deficient GISTs. So before I end, I found 2 more posters during ASCO, which I think is worth highlighting for the GIST community. And the first poster was presented by Dr. Zhang and colleagues from Guangzhou, which is actually a real-world evidence amalgamation of their clinical data of patients who have been treated with avapritinib as well as sunitinib, and GIST patients who actually have failed standard treatments. And this is the early results of their multi-institutional study. And essentially what they have done is actually they have looked at patients between January 2022 to September 2023. And in the patients that they treated, they were given avapritinib in combination with sunitinib continuously for 28-day cycles until disease progression or discontinuation. So you can see initially there were 25 patients, and in actual fact 20 patients were available to be used for analysis for this particular cohort. Now, in terms of results, what you can see is out of these 20 patients, many of them were heavily pretreated. Many of them have relatively large volume disease, and amongst them, they had patients who had primary KIT mutations, as well as primary KIT 11 mutations, as well as KIT exon 9 mutations, as well as some resistant mutations as well. I think without going into too much detail, because this was presented as a poster, and hopefully later when they have more data, they'll be presented at an oral session. But just suffice to say that there are evidence of responses in these heavily pretreated patients, again, in the use of a combination of avapritinib and sunitinib. Of note is the fact that most of these patients would have had prior exposure to sunitinib, and this is a re-challenge of sunitinib together with avapritinib. And you can see that many of these patients actually had good responses in their tumors. And the side effect profile is what you would expect in terms of the use of combination of sunitinib together with avapritinib, and nothing out of the ordinary, I would say. But this is an interesting strategy, similar and akin to the two trials that were presented earlier, where we're really hoping to increase the coverage of the different mutational aspects of GIST by combining two drugs. Another study that was presented as a poster is of a slightly different theme, and this has to do with monitoring through circulating tumor DNA in patients who have had resection of their GIST tumors, which were deemed to be high risk, stage 2 or stage 3. This study comes out of Beijing in China as well. And essentially what they've done is they have recruited patients who have high risk GISTs, who have had a surgical resection. Before the surgical resection, they would take some blood for what we call a ctDNA analysis to look at the circulating tumor DNA within the body. At the time of surgery, of course, some of the tumor tissue would be banked as well as analyzed, and then they would take blood at various time points throughout the patient's journey during the adjuvant therapy phase, as well as at the same time at the different times of imaging as well. And as you can see here, this is the blood taking schedule, and the patients with their tumors, the tumors would then be subjected to whole exome sequencing. And essentially the tumors there'll be mutations found in the tumors where each of the patient would then have a personalized panel, really to look at what mutations their tumors carry, and then actually going back to the patient, looking at the bloods to see whether or not these particular mutations are present in the circulating tumor DNA throughout the patient's treatment journey. So it's a bit complicated, but essentially using a bespoke way of analyzing whether or not there is the presence of circulating tumor DNA in that particular patient. And without going into too much details, just they've highlighted one or two cases who have gone through this process, and what they really want to bring home to the community is that they have managed to use this particular method to preemptively predict whether or not the patient will have disease progression. This is one of the cases which they shared where initially actually the circulating tumor DNA, and so their blood test was positive, but then during the treatment, in actual fact, they were negative. And at the time here, whilst the patient is actually on sunitinib, it became positive again. And at this time point at M9, in actual fact, when they were doing the imaging, they did not see a tumor, but they found that in the blood it was positive, which later subsequently imaging showed the fact that there was evidence of recurrence. So essentially saying the fact that using this particular approach, you're able to preemptively detect patients who will subsequently have what we call radiological progression, and potentially knowing this a bit earlier may allow you to change the treatment of the patients earlier as well. And hopefully with that approach, you will be able to change the outcomes of patients. Now, in conclusion, I think it's been a long 40 minutes. I do apologize for going a bit over time, but I think there are multiple efforts ongoing right now to tackle the issue of treatment resistance, either using novel drugs or a combination of therapy. And as we have more drugs becoming available, we do need these clinical trials to really allow us to test whether or not these approaches are helpful and useful, and also through clinical trials to benefit our patients. We're awaiting further results with larger patient numbers to confirm the efficacy of these approaches. The abstracts I highlighted are mainly in the phase 1 setting, so we do need the larger trials to support. We're also looking forward to the Cogent Peak study, the phase 3 study to read out, hopefully to give us some direction. And the last abstract, which talked about the use of circulating tumor DNA, which I believe I've talked about in the previous webinars as well, which is a bespoke tumor-informed approach, may allow us to predict whether or not patients are responding, or preemptively be able to change the treatment in those who are not responding. And we do, again, need more trials in the future. And there are more trials ongoing, and they will be read out. So I think with this, I'll end, and I'll be happy to answer any questions or clarify any of the points that you might want to raise. So thank you very much.
Thank you. A great overview that you provided.
Sorry, one last shout out. I forgot, this is one more slide. The last shout out is our upcoming sarcoma meeting is in November. It's the CTOS meeting. Wow, it's already been almost a year. So we look forward to more information being presented there as well. So, and it will be in the US. So for those who can come, we also have special registration rates for patients and patient advocates. So certainly try to make your way to the sunny West Coast if you can, and that'd be great. So thank you.
Amazing, thank you. And for those who are sort of new to our webinars, Dr. Loong has also presented updates from our CTOS meetings in the past. So thank you for pointing that out, where it's a little bit more focused on the sarcoma space at this meeting. But for now, given that we're now sharing about ASCO updates, please feel free to enter your questions in the Q&A feature. There is a question that somebody asked regarding reading in Bloomberg that Takeda Pharma has already signed an option to market olverembatinib outside of China. This patient's an SDH patient, so curious about any insights on what does this mean for U.S. patients for clinical trials in the U.S. and or FDA approval, fast tracking, as well as any off-label use if it's only approved for leukemia, as you've mentioned before. So what's its applicability in the future?
That's very interesting. That's a very, very good question to begin with. And I'm only learning this from your question as well. I did not know that Takeda has taken that option, which is, I think, good news. Definitely, it would hopefully, number one, improve access for the use of olverembatinib outside of mainland China. And I believe that's the only place right now where the drug is approved. Now, having that access, I believe, again, the indication is for CML, but it also means that potentially there can be patient access programs or specific single patient cohorts or single patient programs, which can be rolled out in other jurisdictions as well. That really has to do with, I suppose the decision has to be, the decision rests on Takeda or whichever company which is managing that drug, and also to see whether or not they are hoping to then also collect the data to seek for, say, regulatory approval as well. The different regulatory rules and regulations in different jurisdictions are quite different. So I'm not completely familiar with, say, the US FDA in terms of what they would accept. But for a drug that typically is already registered for one indication, it should be quite feasible to open up a compassionate access program for another indication, at the same time trying to collect information which could then subsequently lead to the approval of that drug, especially in a rare disease. And certainly sdh-deficient GIST would be classified as a rare disease by the US FDA. Again, I think the GIST community should try to lobby for access for olverembatinib. Certainly out of all the other compounds that have been investigated in sdh-deficient GIST, this seems to be the most promising one. And because of the fact that there is already a good collection of information in terms of, say, safety data in all the patients that's been treated with the drug because of CML, I think the pathway to having patient access of olverembatinib in sdh-deficient GIST should be quite easily navigated. if it's done properly. So good news, and I'm happy to find out a bit more and come back to the group later with some more information if I am in possession of any.
Okay, great. Thank you for that. Well, we already started off with a great question. Feel free to also, for others, to start entering your questions in the Q&A. You had mentioned earlier about a single arm open label phase 3 and to maybe describe what does that mean compared to other types of trial designs, if you can
Yeah,
elaborate more on
so
that.
sure. So now I don't have the full details. That was one of the Q&As that were asked actually at ASCO. And some of the slides that you saw were actually my discussion points because I had the pleasure of discussing some of these. And to begin with, again, the question is it really worthwhile doing a large phase 3 study, typically, which means that you're comparing with the standard treatment? But to begin with, there is no good standard treatment for sdh-deficient GIST, so it'll be very hard for you to find a comparator. You could compare with placebo, which essentially means it's a non-active compound, but at the same time, it's also not fair for patients when you know that there is so much promising activity in this drug over a sugar pill, for example. So a non-randomized open label phase 3 trial essentially is a trial where the endpoints, so the specific question that's being asked is quite clear. The question is it better than or is it efficacious? And in actual fact, there is no comparator. And I think the reason why they do necessarily need to move on to this trial is when they first designed the trial that was presented by Dr. Chiu, although he only presented the sdh-deficient GIST cohort, that trial initially included all GIST patients. And then the groups with KIT mutations were subsequently stopped because it was not efficacious. So in terms of the size, the sample size, or the statistical power to prove whether or not the drug is useful is not actually fully, it's not ready, or it's not adequate to prove its efficacy in just the in just the sdh-deficient group. So they do have to run a new clinical trial. But I would imagine that the design will be very straightforward. So it would be essentially patients with sdh-deficient GIST and just going over a period of time. And I think at the same time, what they'll do is collect real world data. So data of patients who also have sdh-deficient GIST, but for one reason or another is not on the clinical trial, to see what their trajectory is like in terms of their disease control, and also to compare the patients who are on the trial. And having this control, what we call synthetic control, will be able to give us more information.
Thank you. I'm waiting for any other questions to come in. We have Dr. Loong's time today, so feel free to type in any questions. Perhaps you covered all oh, there we go. Okay. I don't think we have there's a question about just following up on the first question about just connections with, carrying out ovenratinib through any of our physician network. And I will have to sort of look at that offline to see if that's a possibility to learn more. Well, perhaps Dr. Loong covered all his bases here and that you guys feel satisfied with the presentation and it informed you about all the updates so far in the GIST research clinical trial space. He does make himself available if there's any other follow-up questions that come up that people might not think of right here on the spot. So we appreciate you always making yourself available to the community. I think with that, I guess we can give people a little bit of an earlier, end, some more time to your day or evening. And I just want to thank you for always being so generous with presenting to our community and partnering with us. And thank you to everybody here today who listened in and will be listening to the recording, as this is important for our patient community to stay abreast of like what updates there are in the research and trial space. So with that, thank you very much for your time, and everybody have a good day. Be well.
Thank you.
Bye.
Bye.